I do have disclosures, although I don't think they're terribly relevant to today's topic, but I'll disclose them anyway, that I am on the Scientific Advisory Board of OPCO Renal, and I also am on the advisory panel for DQAS, which is the policing organization that studies assays around the world. So I'm here to provide an update on both the enzyme that I'm going to talk about, which is CYP24A1, which you may know, if you're endocrinologist, as the 25-hydroxy vitamin D3 24-hydroxylase. And also to provide an update on the disease that we've now discovered that is related to CYP24A1, which is idiopathic infantile hypercalcemia. Now let me say right off the bat that not all idiopathic infantile hypercalcemia is caused by defects in CYP24A1, but certainly a major proportion. We'd estimate maybe 35% of IIH, I'll call it, is caused by CYP24A1 mutations. And I'm privileged as a biochemist to be associated with several of the studies that have come out on this. So I'm going to give you an update on this story. It began about 6 years ago when some German pediatric nephrologists contacted me about some patients that they'd found that they suspected of having hypercalcemia due to aberrant vitamin D metabolism. And I was pleased to be able to help them because I'd been studying CYP24A1 for about 30 years and had much of the, let's say, the laboratory material that enabled us to study this problem. Let me say right off the bat also that we were trying to prove that the idiopathic infantile hypercalcemia that they were observing in their German patients was in fact due to defects in CYP24A1. It was not obvious because there are polymorphisms of this gene as well as mutations. So this disease for the clinicians is characterized by laboratory findings of a hypercalcemia, which you see over here on the right panel. It's also accompanied by low PTH, really suppressed PTH, which is not uncommon in other types of hypercalcemia. But what you actually see is an aberrant vitamin D metabolism, which we'll talk about in a second. And also you see hypercalciuria, nephrocalcinosis, and also nephrolithiasis. So inherently, this problem is more interest perhaps to nephrologists who keep seeing renal stones and don't know what is the cause. So we're talking now that this is actually connected to an endocrine problem. And that endocrine problem is the fact that there are loss of function mutations in CYP24A1, which you see plays a major role in the catabolism of vitamin D, either the substrate 25-hydroxy-D, the circulating form, it's not being adequately destroyed, or the hormone 1,25-dihydroxyvitamin D3 that you've already heard about from Chantal is not destroyed to calcitroic acid in these individuals. So the result is both elevated 25-hydroxy-D and also elevated 1,25-dihydroxyvitamin D3, and that these two metabolites are therefore altered in this condition. The result is accelerated actions of vitamin D through the vitamin D receptor in all of the biological effects of vitamin D, from the classical calcium phosphate conditions to the types of effects that Chantal was mentioning in the first lecture. So if we study CYP24 in a bit more detail, we see that it fits into the overall metabolism situation quite well. You see all the orange arrows here indicating the action of CYP24A1, and it gets no respect, this cytochrome, because we tend to focus, as Chantal showed in one of her opening slides, on the activation of vitamin D to 125D, and believe that that is sufficient to explain vitamin D metabolism. As is shown in this condition, it's not. CYP24A1 plays an important role in clearing the substrate to 2425D, or the hormonal form 125D into either of two different pathways. One makes calcitric acid, much more common in humans, or into a lactone that's formed in certain other species, such as rodents. So we have, curiously, a cytochrome that can perform two different hydroxylations, one at the 24 position that we see here, and one at the 23 position. We don't know why animals would have both of these pathways, but we do know from lots of other endocrine situations, like in the adrenal, that cytochromes tend to do more than just a single hydroxylation step. They'll often multi-hydroxylate their substrate. So this is classic cytochrome P450 work. We now know a lot about CYP24A1, largely because of the work of Andrew Analora, who published this crystal structure of rat CYP24A1 about 6 years ago now. You can see the helical structure. There's 500 amino acids in this particular protein, and in the middle of it is the heme binding group that's responsible for the catalytic activity that does that hydroxylation step. Now, over on the right here, you have a model constructed by one of my workers, David Prosser, showing the important residues around the active site, around the vitamin D molecule, that are responsible for holding this molecule in positioning the side chain just right for 24 hydroxylation or 23 hydroxylation. Interestingly, to illustrate this residue AL326, if we modify that to a glycine, we can change even the position of the hydroxylation from 24 to 23. So it shows this model's not a bad model for indicating how vitamin D binds. Recently, I've had the chance to look at some cells that have overexpressed CYP24A1 with a green fluorescent protein tag. And here you see that protein showing its green fluorescence, and you can see that its expression coincides inside the cell with the mitochondria, not with the nuclei, which are blue. When you actually put all of the dyes together, you see this rather pretty picture of mitochondria containing CYP24A1. So this is a mitochondrial P450, not a microsomal one that you're perhaps more familiar with through drug studies. So in that work on IIH with the German pediatricians, the first thing we wanted to do was to prove that the cytochrome that was affected was not simply a polymorphism, but actually a mutation. So what we did was to take the wild-type human CYP24 and reintroduce the mutations that were found in people and see if, in fact, they alter the activity of this CYP24. And to do this, we expressed the cytochrome in a V79 host cell, and then incubate with a 125 dihydroxyvitamin D3 to see what the products would be. We were expecting to see then the familiar catabolic products, 124-25 for instance, and not see them if the protein was mutated in a crucial way. We assayed the enzyme and studied it by a combination of radioactive substrates, HPLC, and also by LCMS-MS. So this shows you what happens when, in that study that we published in the New England Journal, where we study radioactive 125, there's a nice big peak of 125 dihydroxyvitamin D3 that's unaffected in the host cells. And then when we put in the wild-type CYP24, we see metabolic products. The 125 dihydroxy-D disappears. In all of the mutant types, and you see 4 of them here, including this very common E143 deletion, you see that the 125 remains after incubation, and there are no metabolic products, proving that these are loss-of-function mutations and not polymorphisms. In more recent data that we've been acquired using LCMS-MS, we can study the first product on that metabolic pathway, 12425, by studying the mass signal, and we can, in this particular case, it produces 2 peaks, and we can study in the red peaks the wild-type enzyme, and you see in various different mutant forms, several that are approaching the wild-type activity, and then many down below here that have completely lost biological activity. So this is another valid way of studying the enzyme using LCMS-MS. Of course, this is a bit laborious if you want to study these patients quickly, like the clinicians do. And so to be able to distinguish between, as is shown here, several mutations and the various polymorphisms, we needed a more rapid method to be able to distinguish these patients. We've now studied about 20 to 25 different mutations that can cause this IIH condition, and studied these all using our in vitro technology, but we're still after this fast method to be able to distinguish these people from polymorphisms, and so we came up with a method. And that method is based upon LCMS-MS. You hear a lot about it. In fact, there's a symposium this afternoon about LCMS-MS. In comparison to radioimmunoassay, this can now be extended to vitamin D. We have a method which works on small volumes of serum, that works on, it's very sensitive. It allows us to measure several vitamin D metabolites at the same time. And so when you hear about vitamin D assays, just remember that LCMS-MS is rapidly coming along and replacing the radioimmunoassay as the method of choice, because it allows us to measure lots of different metabolites simultaneously. This particular approach that we use involves a dienophile, a chemical agent, to bind across the cis-triene of vitamin D and therefore tag all the vitamin D metabolites at the same time. The eventual sensitive assay allows us to look at several aliquots of 17 microliters of serum in a single run. When we apply this method to regular people, so these would be in case of about 700 women studied in Nebraska, we can see that there's a relationship between 25-hydroxy-D levels and 24-25-D levels, which is a metabolite we're really interested in as being a measure of whether a patient has IIH. So the first problem we have, of course, is to say, well, it doesn't go through 0. And that's actually related to the previous talk, would you believe, that we can define vitamin D deficiency in several different ways. The most common way that clinicians use is to use the level of PTH as a measure of vitamin D deficiency. As PTH rises, it's telling us that the vitamin D metabolism is stressed. But another way that we know that vitamin D metabolism is stressed is when the body turns off the production of 24-25, as is shown here, at about that level of vitamin D deficiency that we were just hearing about, 25 nanomoles per liter, that we can see here. So this is a very good measure of determining when somebody's vitamin D deficient. So the first problem then is there are people who have very low 24-25. We don't want to have those confusing our studies of IIH, the hypercalcemic condition, and they don't, as you'll see. Now this is the biggest group of IIH patients that have been assembled so far by a Dr. Kotler in France, in Cannes, France, and she has assembled 18 of them, which of course she sent the blood to us for analysis using our new method. And we can see that these patients, they are hypercalcemic. This is the group B that we're looking at. They have low PTH, and sometimes they have high 125Ds, but not consistently. It's not a great measure of hypercalcemia, is the 125 at the time of analysis. On the other hand, 25-hydroxy-D is elevated in these patients, not low. 24-25 is virtually unmeasurable. And so what's become the reliable indicator of IIH is the ratio of 25-hydroxy-D to 24-25D. And we can see this becomes in the hundreds, whereas in you and I, it's usually between 5 and 25. So it's quite a useful measure of IIH is this ratio. And that has been found in all of her patients. And I can now extend this to include Dr. Schlingmann's original patients from Germany, and now there are 28 patients in this study, including the Kotler and Schlingmann patients. And we've modified our method slightly and increased this ratio that we see by cleaning up the peak of 24-25 slightly. And the outcome of this is that all these patients with IIH have elevated ratios of 25-hydroxy-D to 24-25. So if anybody has in the audience a patient who they suspect as having IIH due to CYP24, send me some serum. Be pleased to analyze and tell you whether this ratio is abnormal. In fact, it takes about 10 minutes to do this, whereas those earlier studies doing modifying the gene take probably 2 months. Also of note here is that the heterozygotes, those are the patients, those are the parents of patients, the siblings of patients with one affected allele, do not show hypercalcemia, they don't show the disease, and they don't show this elevated ratio. So it allows us to distinguish between IIH patients and heterozygotes. The other group that we should remember now exists is IIH due to other reasons. And one of these is SLC24A1. These patients have a normal ratio also. And we published on this in January of this year that there is another cause of idiopathic infantile hypercalcemia. It's due to this sodium phosphate transporter, SLC24A1, and that's totally unrelated to CYP24. In this case, it's a change in the FGF23 aspect of calcium and phosphorus homeostasis. Causes elevated 125D, but it doesn't affect that ratio of 25 to 24-25. Going back to that, there it is. There's the ratio, it's quite normal in these patients. So this test then allows us to distinguish those IIH patients who have a defect in CYP24 versus those that have a defect in the sodium phosphate transporter. Over here on the right is the only group that we found that might be confused with IIH. They are dialysis patients, and This is the reason why that they have very low 24-25 levels for a given 25-hydroxy-D. This is 700 patients in data we've acquired with Laura Armas in Nebraska, and you can see that they have abnormally low 24-25, abnormally high ratios, and so they could theoretically be confused. But the two conditions are never seen at the same time usually. So what makes these patients so susceptible to hypercalcemic episodes? I thought I would take time to comment on this. One is vitamin D intake. IH patients should be advised to avoid vitamin D supplements. So although we've heard some advice to normal individuals, these patients should not be given vitamin D supplements. The best example of this is two brothers that were detected in Germany. One brother was detected before the disease was first described. The second brother was detected after the disease was first described. The second patient was never given vitamin D supplements and has never developed hypercalcemia. So increased vitamin D definitely places additional burden on catabolism. The other example is an exception to the second speaker's talk about pregnancy, and that is that female IIH patients do develop hypercalcemia during pregnancy, and now there are at least 3 or 4 examples where females have been shown to do this, and we believe the reason that they do this is that placenta makes 125, we've heard that story today, that additional CYP27B1 is what's putting a particular emphasis on the fact that these patients don't have CYP24A1 to balance that story out. The example I'm going to show you is this patient with a childhood history of recurrent renal stones. She presented with her 4th bout, a 4th episode of hypercalcemia during pregnancy, and she had, as we detected, a very high 25 to 24-25 ratio by our method, and genetic testing confirmed that she had 2 different mutations as a compound heterozygote. Of course, the infant's normal. The infant would be a heterozygote. Now, this has now been, let's say, corroborated by 2 other studies, one by Denor, and there's also a very recent paper in the last month, a similar patient. So they were IIH patients with CYP24 defects. They couldn't handle the recurrent hypercalcemia that they see during pregnancy. Interestingly, these patients are not hypercalcemic when they're non-pregnant. I'll just finish up by just mentioning an animal model that we have for this condition. Now, this is the CYP24A1 knockout mouse, null mouse, from René Saint-Arnaud. We've studied many of these mice in terms of our blood test, and we can say that they have a picture just like the humans with elevated 25-hydroxy-D, a very low 24-25, and a very elevated ratio that we see also in the human. Interestingly, since René Saint-Arnaud developed that mouse, Wes Pike, the University of Wisconsin, has taken this mouse and reintroduced the mouse and human genes for CYP24A1 to create a knock-in mouse. And this mouse has been studied both for its phenotype and for its vitamin D metabolism by us. And interestingly, these animals are rescued by the knock-in gene. It can be the mouse or the human. The data are quite similar. Just to remind you that the mouse is able to make galactone as well as 24-hydroxylated metabolites like calcitroic acid. So it's a bit more complicated than the human story, but more interesting for us, the analysts. And I'll show you some of the raw data. Here's the normal mouse in red. It's a heterozygote. It has normal 25-hydroxy-D, normal 24-25-D, and normal lactone, another metabolite that's made by CYP24A1. When we create the knockout mouse, we actually have elevated 25-hydroxy-Ds, probably about 10 times normal. We have hardly detectable 24-25 and virtually undetectable lactone. When we rescue these animals with either the mouse or human gene, 25-hydroxy-D in green goes back to normal, 24-25 is actually supernormal, and the lactone is returned as well. So these animals recover their abnormal vitamin D metabolism by this gene approach. And that actually shows some promise, because this is with the CRISPR technology, that patients with this problem could technically be given eventually the human gene again, normal human gene. So I say future directions of consortium. I work with a group of clinicians and with a group of people who study the mouse, and we're studying various aspects, in particular to see if there are additional defects of vitamin D metabolism in hypercalcemic patients. We plan to continue testing those various mutations. And also, quite interestingly, René Saint-Arnaud again has created several knock-in mice also that have the human defect reproduced in an animal model. And these include this mutation called R396W, which is quite common around the world. So in conclusion, I've shown you that CYP24A1 plays a crucial role in clearing vitamin D. When we don't have it, there's consequences, severe consequences of hypercalcemia, and I hope that eventually it will gain a prominence that's seen for the 1-alpha hydroxylase also. Thank you.